conference and you give lectures, you always express your gratitude to the organizers. But I will not do this because I am a part of the organizer. So I can't say that I give gratitude to myself. So I am the co-organizing secretary of this conference. So just a few things I would like to say, and a big round of applause for Prof. Sudhir, my teacher, and he was also Vice Chancellor of Lucknow University, also occupied the chair of Vice Chancellor of Lucknow University. And Dr. Kusum Yadav from my department, and Sanjay Yadav is her classmate. They are here, and I believe that it's God's will that they are in the nick of time before my first slide begins. So, a bit thing about myself. I am professor of biochemistry at the University of Lucknow. Lucknow University is a 102-year-old institution with the story starting. Namaskar. With the story starting on 25th November 1920, and very recently you all must be knowing the Lucknow University got A++ grade on July 26, 2022. So, big round of applause for Lucknow University. But that was a historical occasion, and never ever a state university got this distinction. So, actually, my topic of the lecture is cancer as a lifestyle disorder. So, what causes lifestyle diseases? There are certain habits, behaviors and practices such as poor eating habits, inactivity or smoking and drinking. Factors which we can easily control. So, actually, cancer has spread its tentacles like an octopus. So, it is very important for us to spread awareness about cancer. And cancer is increasing in the developing nations of the world. Cancer is increasing in the developed nations of the world. Factors that we cannot control, such as age, such as genetics, such as gender and heredity. So, this is a famous quote by Mickey Mantle. If I knew I was going to live this long, I would have taken better care of myself. Now, who is Mickey Mantle? He is an American professional baseball player. Dr. has come all the way, he is there. Dr. Sethi has come all the way from America. Baseball player. Mantle was one of the best players. As you know, cricket is very important game in our country. So cricket is a religion in our country. In the United States, you don't play cricket, you play a game which is close to cricket, that is baseball. Here also you have got a bat and ball, and Dr. Saxena in the beginning of the slides showed a bat and ball. So he was one of the best players and regarded as many as the greatest switch hitters in baseball history. Those who have got some interest in baseball, they must be knowing. And he died of liver cancer at the age of 63 years. Mantle took his first drink. He started drinking alcohol at a very young age of 19 years. In 1994 January, he was told by his doctor that his liver was so badly damaged from almost 40 years of continuous drinking that his next drink could be his last drink. He admitted that he had been cruel and hurtful to his families, his fans, his friends because of his alcoholism. So my lecture is cancer and lifestyle disorders. There are so many parts of cancer which are easily preventable. Doctors discovered that Mantle's liver had been severely damaged by both alcohol-induced cirrhosis and hepatitis C. They also discovered that he had an inoperable liver cancer known as undifferentiated hepatocellular carcinoma. So I begin my lecture by giving popular examples of celebrities. You all know this. You all know this. Superstar Vinod Khanna. You all know. He is such a superstar. He was a ruggedly handsome man in life, shirt and button, muscle rippling. So many pictures like Amar Akbar, Anthony, Parvarish, so many pictures you come to your mind. Cigarette dangling rakishly from his lip. He was unrecognizable in death. Can you believe that this is the same man who is standing here in his young days? Shashi Kapoor, Rishi Kapoor, Irfan Khan, Rajesh Khanna. They were celebrities and they all died of one common disease, that is cancer. Vinod Khanna, one of the last esteemed titans of a generation, battled a lethal form of bladder cancer, resistance to chemotherapy for 6 long years and finally succumbed on April 27. You all know Rajesh Khanna was the first superstar, and you must have seen his movie where he was waving to his fans just before his death. He died because of liver cancer. और अगर आपने राजेश खन्ना को अपने हैंडसम यंग डेज में देखा हो और राजेश खन्ना को अगर आपने उस फोटोग्राफ में देखा हो, जैसे चूसने वाला आम होता है ना, जब उसको आप चूस लेते हैं, उसकी जो स्थिति हो जाती है वही कैंसर स्थिति कर देता है इंसान. So it's the best of times It is the worst of times on the cancer front. Scientists continue to be baffled by the complexity and smartness of cancer cells. They find ways to dodge even the most powerful therapies. It's a war between the cancer biologists and cancer cells. But not hundreds of distinct diseases. That each individual cancer behaves differently. That two people with the same cancer at the same stage receiving the same treatment can experience radically different outcomes. So the cancer cells, they always find a strategy to evade our immune system. Cancer is catching up with the heart disease as the leading cause of death globally, reports the World Health Organization. What causes cancer? Cancer is caused by alterations or mutations in the genetic code. It can be induced in the somatic cells by chemicals which induce cancer, known as carcinogens. Radiations. Chemicals are used for the treatment of cancer in chemotherapy, which I'll talk about later. Radiations, they cause cancer, and radiations are used for the treatment of cancer in radiotherapy. This is the paradox. There are certain viruses. There are certain cancer-causing viruses. DNA cancer-causing viruses, the examples of simian virus 40 and Rau sarcoma virus. Heredity, 5%. Cellular basis of cancer. Cancer is characterized by abnormal and uncontrolled growth of cells. Cancer arises from a loss of normal growth control in cells. As you all are aware of the fact that plant cells, there are certain plant cells like apical meristem cells which keep on dividing till the plant is alive. But in animals and humans, the cells, they stop dividing after a particular age, except for during what you call wound healing. The cells do divide. Otherwise, there are regulatory constraints. In cancer, this balance is disrupted from uncontrolled cell death or loss of cells' ability to undergo apoptosis. Apoptosis, as you all are aware of the fact, apoptosis is programmed cell death. It is brought about by enzymes which are known as procaspases, which become activated, they form caspases. There are initiator caspases, terminator caspases. Any cell can be killed by apoptosis. Properties of cancer cells. Cancer cells, they become immortal. The person dies, but the cancer cells become immortal. Cancer cells often avoid senescence or cell death by maintaining their telomeres despite repeated cell divisions. Now, this is possible because cancer cells express high levels of telomerase activity. In normal cells, the telomeres, they shorten and finally it comes to a stage when they become so short that the cell dies. But the cancer cells, they express high levels of telomerase activity, an enzyme which adds genetic units to the telomeres to prevent them from shortening to the point causing senescence or cell death. High telomerase activity enables the cells to divide indefinitely. Telomerase is active in 85 to 95% of cancers. Cancer cells have got certain special features. They express certain antigens which are not expressed by the normal cells. Cancer cells हमारी body में कहीं बाहर से नहीं आते. It is the normal cells which get transformed into cancer cells. All of us who are sitting in this auditorium cannot say that we will never develop cancer in our lives, because we all have got cancer-causing genes which are known as oncogenes. And these oncogenes are present in an inactive form which are known as proto-oncogenes, and when there are certain factors which trigger the conversion of proto-oncogenes into oncogenes, then cancer is caused. Lack of substratum. Now, you know, cancer cells, they do not require an anchorage or substratum. Then loss of contact inhibition. Normal animal cells, when they come in contact with a dividing cell, when they come in contact with another cell, they stop dividing. Cancer cells have got no such inhibitions. They have got a reduced requirement of growth factors and angiogenesis. Since, you know, in a normal cell there may be 10 cells. When cancer cells are there may be 10,000 cells. So they require extra blood supply. Because blood is required for oxygen, blood is required for nutrients and other things. So they have requirement of new blood vessels, and this is a phenomenon which is known as angiogenesis. A tumor is an abnormal lump or growth of cell. Tumor is of two types: benign tumors, which have got a localized existence and they do not pose threat to our existence. So normally when biopsy is done, if it's a benign tumor, it doesn't pose threat to our existence. Otherwise, if there is a malignant tumor, it's a neoplasm, then it comes into blood supply, then it goes to different parts of the body, and then they can establish new cancer at new places. Biopsy, you carry out fine needle aspiration therapy Early diagnosis is key to the successful treatment. I will skip this slide. Now, nucleotide changes in the genome of cancer, which is the unseen side of the iceberg. You know that iceberg word has been used specifically out here. You know, iceberg is there in the sea, and you only see the 1/10th portion of the iceberg. Titanic is a movie, you all know that the ship Titanic, it hit the iceberg and so many people died. So, major portion of the iceberg is inside the sea. So, nucleotide deletions are there, nucleotides are deleted, nucleoside insertions are there, new nucleotides are inserted, nucleotide substitutions are there. So, mutation is the basis of cancer. Normally, body have got DNA repair mechanisms, and the body takes care of the DNA damage which takes place continuously. As we age, the rate of accumulation of mutations, it far exceeds the rate of DNA repair, and that's why normally we get cancer at an advanced age of across 60 years. What are the genes which are responsible for tumorigenic cell growth? Proto-oncogenes, I just now told you, all of us they have got proto-oncogenes, and they have got a loss of function mutation. It leads to uncontrolled cell growth and cell proliferation. Proliferation means increase in size. Tumor suppressor genes are there. Caretaker genes are there. These are the genes which protect our genome from cancer. And there was a loss of function mutation. And mutated or activated oncogenes, they got malignant transformation, loss of gain or mutation of tumor suppressor genes. So it is the entire story is in our genome. These are the oncogenes. There is a whole family of oncogenes. RAS is there, FOS is there, MYC is there, SRC is there. Now, this is the proto-oncogene RAS. All of us they have got these oncogenes. And this RAS, when it is associated with GDP and it is in an inactive state, when it is associated with GTP, it becomes activated and then it causes protein products, and these protein products, when they are expressed, and cancer cells have got an altered surface morphology. They express certain antigens which are not present on the normal cells of the body. There are two categories of antigens. One is tumor associated antigens and the other are tumor specific antigens. So, now here what you are seeing is that this is the RAS gene, and these are the amino acid positions when the A product form the protein products. At the 12th position, 59th position, and 61st position. So CRAS, HRAS, KRAS, NRAS, HRAS is the RAS family. And here with the blue color, you see that these amino acids they have been substituted. Glycine, this is substituted with leucine. You have got this is in the normal cells. Here you have got lung carcinoma, bladder carcinoma, and cysteine out here. You have got lung carcinoma, colon carcinoma. So amino acid substitutions they trigger the formation of different types of cancer. So the result is overproduction of growth factors, flooding of cells with replication signals, uncontrolled stimulation in the intermediary pathways, and cell growth by elevated levels of transcription factors. So tumor suppressor genes and caretaker genes, they are responsible for our genomic stability. They play an important role that we do not get cancer. So there is a loss of function in the tumor suppressor gene. Normal function is that they inhibit the cell proliferation and cell division. Absence in inactivation of inhibitor that leads to cancer, and both the gene copies must be defective. P53 is a pro-apoptotic protein and it plays a very important role in preventing our genome from causing cancer. And damaged DNA, it activates this ATP, it's an energy active process, ATP is converted to ADP. P53 undergoes degradation, here it undergoes phosphorylation, and when it undergoes phosphorylation, then it undergoes transcription, DNA undergoes transcription, and it cannot phosphorylate RB protein and the cell cycle is arrested. So this is in the normal person, so that the cancerous growth is prevented, or else it triggers apoptosis and cell death. When these mechanisms they fail, then it leads to cancer. DNA repair genes. So the DNA repair genes are very important, and mutations in the DNA repair genes they lead to an increase in the frequency of mutations in other genes of the body in the somatic cells, such as proto-oncogenes and tumor suppressor genes. Proto-oncogenes they have got a gain of function mutation, tumor suppressor genes have got a loss of function mutation. Breast cancer susceptibility genes, BRCA1, BRCA2, as you all know, that breast cancer is one of the most, ovarian cancer, breast cancer, they are one of the most common cancers in females, and hereditary non-polyopsis colon cancer susceptibility genes. They have got DNA repair functions. Their mutation will cause tumorigenesis, that is the formation of tumor. Importance of DNA repair. So this is the balance. एक तराजू का पलड़ा है, यहाँ DNA damage हो रहा है, यहाँ DNA damage हो रहा है. Here the DNA damage is balanced by DNA repair, and this leads to genetic stability. But here the DNA damage is defective, there is incomplete, there is mutation in the genes which are cause DNA repair, there is mutations in the enzymes, genes which code for the enzymes involved in the DNA repair. This leads to genetic instability. Genetic instability is the hallmark of cancer. You have got cancer and hereditary diseases and genetic divergence. ये तराजू का पलड़ा है, जब तक ये तराजू का पलड़ा इधर वाला झुका रहता है, it leads to genetic instability and we are protected from cancer. And the moment this तराजू का पलड़ा इधर झुकता है, we become prone, we become vulnerable, we become susceptible to cancer. Causes of cancer. Main risk factors, and they are preventable causes. So you are young people and you must take care of this. Tobacco use, sunlight exposure. Sun is the source of light. But sunlight has got UV radiations, and UV radiations they can cause skin cancer. There is a type of skin cancer which is xeroderma pigmentosum, and where the skin becomes very sensitive to sunlight. The source of UV light on the earth is sunlight. Excessive alcohol use, alcoholism. Rishi Kapoor and Rajesh Khanna and Vinod Khanna, one of the triggering factors was rampant alcohol. They drank alcohol. Rishi Kapoor had a big bar at his house. Rajesh Khanna also. Ultimately, that led to that situation. Getting older, as we age, the mutation rate increases. Family history, cancer has got a very strong genetic factor. There are certain cancers which run in families where even cross 20 years of age, people develop cancer. And genetic factor we cannot control. Poor diet, I'll come over this. Lack of exercise, physical inactivity, sedentary lifestyle, laziness, infection from certain viruses. There are certain cancer-causing viruses. Occupational exposure to chemicals, and chemicals which cause cancer, they are known as carcinogens. So these are the things, and majority of these things, they are preventable. We can protect our genome from ourselves from causing cancer. और मिकी मैंटल ने कहा था कि I did wrong thing. जब उसकी डेथ करीब आई, तो उसको लगा कि मैंने बहुत गलत काम किया, but by that time it is too late. Always better to take precautionary steps. So causes of cancer, genes and cancer, randomly acquired through errors in DNA replication. Inherited and thus present in all cells from birth. The heritability of cancers are usually affected from complex interaction between carcinogens and host genome. Nobody can say that we will never develop cancer. We have got probability of developing cancer, but by having a disciplined lifestyle, we can reduce that possibility to a minimum. Carcinogens such as tobacco smoke, radiation, chemicals, or infectious agents are involved in cancers. Cancer and environment. DNA repair, food, nutrition, obesity, and physical activity or inactivity. Proliferation, hormones. There are many hormones. The imbalance of hormones leads to cancer, particularly breast cancer. Differentiation, inflammation, and immunity. Immunity, if it is compromised, then can lead to cancer. Apoptosis I have already discussed. Cell cycle I have discussed. Carcinogen metabolism. There is a topic which I teach in biochemistry, xenobiotic biotransformation. There are phase 1 reactions, there are phase 2 reactions. In normal toxicants, the toxicity is reduced. But there are cancer-causing chemicals which are known as carcinogens. When they enter into our body, they are in the form of procarcinogens. And the phase 1 reaction, the monooxygenases, mixed function oxygenases, hydroxylases, they convert them into the carcinogenic form. So the earlier term which was given was xenobiotic detoxification. But after, okay. My lecture started 10 minutes late, so that's why I'll take 10 minutes more time. And I am a part of the organizing committee, I will take a bit more time. So, 5 more minutes. Cancer is a comorbidity in COVID-19 infection, so I have been asked by the chairperson. So, we very recently we had a deadly COVID-19 pandemic, and there were 6 comorbidities. Hypertension, cardiovascular diseases, cancer, diabetes mellitus, chronic obstructive pulmonary disorders, and cancer and asthma. And these were the people whose immunity got compromised because of the non-communicable diseases, and they succumbed during this second deadly wave of Delta variant of the COVID-19 virus. So I am skipping certain slides. Now tobacco is a very important form of causing cancer. So this is cigarette, and cigarettes, it has got nicotine, which is highly addictive and contains over 19 carcinogens and 4,000 chemicals. Please avoid tobacco. Please avoid smoking in any form. And tobacco is the leading cause of cancer.
It causes submucosal fibrosis or oral cancer. 70% of the patients are of young age, and 30% of the patients of oral cancer, they come to the hospital in the late 4th stage. For the cancer treatment, early detection is the key. Early detection. In our country, people go very late to the doctors. So tobacco products, Khani is there, BD is there, people are very fond of BD, cigarette is there. You know, smoking of cigarette is taken as a feeling of machoism. Gutka, chillum, mawa, snuff, gutka, pan masala. So these are all very dangerous things. So tobacco in India, 5 crore people, they earn their livelihoods to tobacco industry, which is flourishing but raises huge concern to the health of the Indians. 6,000 crores of foreign currencies earned by the Indian government. So alcoholic drinks, another very important precipitating factor for tobacco is alcohol, and it causes depression, it causes memory loss, oral cancer, breast cancer, impotence, impaired sensation leading to falls, लोग ठीक से चल नहीं पाते हैं, all these sorts of things are there. UV radiations have got beneficial aspects also. They are shown by green color, killing of the pathogens of the skin, vitamin D synthesis, you know, UV radiations they play an important role, treating disorders, but they also can lead to edema, to skin damage, DNA damage, cell cycle arrest, apoptosis, genetic mutations, and ultimately leads to carcinogenesis. So radiations, they cause cancer, and radiations are also used for the treatment of cancer. Now alcohol, smoking can cause 14 types of cancer. Third, lung cancer. Smokers are 23 times more susceptible to develop lung cancer. Smoking is perfectly preventable. I request to you all, never go for smoking. किसी ने कहा कि एक बार सिगरेट पी के देखो, don't do that. Abstinence is the only way. And you know, they have got carcinogens like DNA benzopyrene, they cause sufficient DNA damage. So I'm quickly skipping these slides. Then alcohol. When we drink alcohol, whether in the form of beer people think of as a cold drink, but beer is also a cold drink, and beer can also, it's an undistilled alcoholic beverage, and wine and gin and rum and vodka and things like that, as the alcohol percentage increases, if in a small amount alcohol is drunk, the body can process it without much damage. It is converted by alcohol dehydrogenase into acetaldehyde. But if we drink in large amounts, then it causes mistakes in DNA, it causes chromosome rearrangements, it causes DNA, binds to DNA and produces lumps. So treatment is radiotherapy, chemotherapy, surgery. I am quickly skipping through these slides. Then mammography, which is a specialized medical imaging to see the inside of breast. Any girl, if at any age, 10 years, 15 years, if she feels a lump in the breast, it should never be removed. That can be very dangerous. And you know, Angelina Jolie was a famous Hollywood heroine, and she underwent double mastectomy because she had a breast cancer. She had a genes which made her susceptible to future breast cancer. There are magic bullets, immunotherapy. There are this monoclonal antibodies which are tagged to radioisotopes. So I'm quickly skipping these slides. One thing which is very important: 12.5% of Indians are diagnosed at an early stage, whereas a cancer can be cured, compared to 86% in USA. USA में जितने लोग डॉक्टर के पास चले जाते हैं अर्ली स्टेज पे, जितने लोग नहीं जाते हैं उतने लोग यहाँ इंडिया में जाते हैं. That's why Indians they succumb to cancer much quickly, and now a lot of things are taking place. जो है gene editing in CRISPR-Cas. Now this is the latest technology. You know, CRISPR-Cas is a system of the adaptive immune system in bacteria, and CRISPR-Cas technology, they are like molecular scissors. You carry out a molecular surgery on genes. These are some of the latest slides which are prepared for this. Now these are the cancer cells. Using CRISPR-Cas9 in the study of tumor therapies. These are T cells. You know, there are T cells of two types, CD4 T helper cells, which are infected by HIV, and CTL, cytotoxic T lymphocytes, which attack and kill the cancer cells. So you use these molecular scissors like Cas9 and CRISPR, and you restore the cell's ability, anti-cancer infection, and then they are treated with these cells. So actually the gene editing and knockout anti-oncogenes. You or any student of immunology, you know, you've got knockout genes. So you knock out the oncogenes, and then you have an animal tumor model, and then you treat the cancer. So these are some of the latest technology. They are still in the experimental stage, but they promise a lot. Intelligent surgical knife. So this, I'm not going into all those details because the paucity of the time. Then one thing which is very important in the end, I will talk about CAR T cell therapy. CAR T cell, the full form is chimeric antigen receptors. CAR T cell therapy is a recent breakthrough innovation in cells and gene therapy. It uses the patient's own immune system cells to kill the cancer cells. Selectively kill the cancer cells. Radiotherapy, chemotherapy, surgery do not differentiate between normal cells and cancer cells. There are no fixed boundaries कि यहाँ से यहाँ तक cancer cells है, यहाँ तक normal cells है. Normal cells are killed. There is collateral damage. Cancer is an entropic disease. To make the CAR T cells, scientists collect the samples of patient T cells from the blood and engineer them to sprout special structures called this. And here you can see in the CAR T cell therapy, you remove the blood from the patient to get the T cells. You insert a gene for chimeric antigen receptor, and this is expressed in the T cells. You grow millions of CAR T cells, and you infuse these CAR T cells into the patient. And another thing which is very important out here is that, and he is the class fellow of Dr. Kusum Yadav and Sanjay Yadav, who are sitting on the front row, and that's why and he is MSc Biochemistry 2001 batch, and he 2000 बैच, तो एक साल मैंने ज़्यादा कर दिया, एक साल अब कम कर देते हैं। ये 2000 बैच के हैं, इन्होंने कंपनी लॉन्च किया इम्यूनो एक्ट। इम्यूनो एक्ट is English binding startup company specializing in CAR T cell therapy. And one thing which I would like to tell you out here, that the same treatment in the United States of America cost 4 crores of rupees, and the cost with Rahul Purwar's company, which was launched in 2022, this year itself, it has come down to 30 lakh of rupees. जिस थेरेपी का ट्रीटमेंट अमेरिका में 4 करोड़ रुपए में होता है, वो 30 लाख रुपए में अवेलेबल है. So this was a slide which I was interested in showing you. These are some of the deadly form of cancer. सिगरेट के पैक पे देखिए क्या क्या चीजें बनी होती हैं. Still सिगरेट का कंजम्पशन बढ़ता जा रहा है. So this is Mickey Mantle. This was in glory days and this was in gory days. तो ऐसी चीजों को अवॉइड कीजिए. अवॉइड किया जा सकता है. 95% चीजें जो है वो प्रिवेंटेबल हैं. So that is all. I have already taken a lot of time, and I express my sincere gratitude to the chairperson for giving me this time. And it is very important for us to spread awareness about cancer, to know more about cancer, because it's an ongoing battle where the cancer cells fight newer and newer strategies to evade the immune system, to cause different type of cancers. But if we lead a disciplined lifestyle, if we avoid tumor, if we avoid tobacco products, if we avoid smoking, if we do a lot of physical activity, then we can protect ourselves from cancer. And it's a dreaded disease. It kills you emotionally, it kills you financially, it kills you mentally. We can always protect ourselves. All of us must have lost some near and dear ones because of cancer. So thank you very much. Thank you.